I'm Tall.
And I'm Small.
And today we are going to be doing Khan Academy's MCAT Prep Biomolecules Practice Questions, Enzyme Structure and Function. I did it again! All right, question number 1. Enzymes catalyze biochemical reactions by altering which of the following quantities associated with the reaction? Is it the activation energy, the equilibrium constant, the enthalpy of formation, or the Gibbs free energy change? Okay, so when you're talking about a catalyst, you're not talking about any of the reactants or products. You're talking about the pathway to get from reactants to products, and that pathway is the transition state, basically.
Perfect, exactly.
And the amount of energy to get to the transition state is the activation energy.
Exactly. So the one thing you don't ever want to pick on this question, because there's a good chance that it could be on there, is the equilibrium constant.
Why is that?
Because if you do pick it, then a chair will fling you out of the testing center. Because that is specifically something that an enzyme never changes. Same thing with Gibbs free energy. Just like you mentioned, we're not working with products and reactants. Therefore, we're not changing the overall favorableness of this reaction. So that was really good things. And then the enthalpy of formation, again, is not something that this changes. We're not changing anything with the reactants.
Right.
So good rule.
Yeah, so activation energy, and sure enough, we got it. Okay, next question. Phosphoglucomutase is a hard word to say.
It's a fun one.
Which catalyzes the formation of glucose 6-phosphate from glucose 1-phosphate. This is a glycolysis question.
Well, not technically.
I learned about glycolysis recently. Is best classified as which of the following enzyme type?
So we get glucose 6-phosphate for when we're chopping up glycogen. So maybe you just went on a run and your muscle cells are saying, oh, we're starving, we need more glucose. And your liver is like, oh, let me chop up some of my glycogen stores and I will feed all of you guys, because the liver does that. He's a really nice guy.
Liver's very nice.
Yeah. So so that's where we get glucose 6-phosphate. But in order for us to use it in glycolysis and get some energy out of it and eventually feed the electron transport chain, we got to convert it to glucose 1-phosphate. So we use an isomerase to do that. And
we could reason this by, if you have glucose 1-phosphate, that's saying that you have a phosphate on the 1 carbon of glucose. Mm-hmm. And if you have glucose 6-phosphate, it's the exact same chemical formula, but now the phosphate is just on the 6-glucose.
Yeah.
So it's, definition of an isomer, isomerase. It was right. Great work. Keep it up for 8 more, guys. All right. Thanks, Sal. Okay. Which of the following molecules cannot be classified as an enzymatic cofactor? Heme, magnesium, FAD, or valine? Cannot be classified as an enzymatic cofactor.
So this is one of those little word games, like a square is a rectangle, but a rectangle is not always a square. So thinking about cofactors and coenzymes. So coenzymes are always cofactors, but not the other way around.
Yeah.
Okay, so coenzymes are always an organic thing that helps the enzyme do its chemistry, basically. And cofactors could be, or they could be something like a metal that's inorganic that helps us do our chemistry.
So the thing that jumps out at me immediately is valine.
Mm-hmm. Yeah.
I feel like valine, it's amino acid.
Yeah.
I doubt that is an enzymatic cofactor.
Never heard it involved in something with an enzyme before.
And I think I've heard of all of the other ones being involved with enzymes.
We flat out know heme is. We know magnesium is required for a lot of DNA binding stuff with enzymes.
Definitely.
And we know FAD is involved in a lot of metabolism.
Okay. Yeah. Valine. Yeah, we got it.
Nice.
All right. Next question. Question number 4. In order to analyze the catalytic effect of 2 different enzymes on the same chemical reaction, it is best to compare which of the following quantities. Okay, so this is the catalytic effect of 2 different enzymes on the same chemical reaction. So we're comparing the action of the enzyme. We're not comparing the reaction.
Yeah.
Like the reactants and products. So is it the difference between transition state energies, the differences in free energy between the reactants and products, the differences in entropy between the reactants and products, or the differences in enthalpy between the reactants and products?
So this is knowing that delta G equals, entropy plus enthalpy, and temperature is in there somewhere, but knowing
There's
that
triangles.
There's a delta in there somewhere, yeah. So knowing that all 3 of those are involved in something that tells us how well that overall reaction is favorable.
Right.
Lets us rule all three of those out, kind of, if that will be the same for both enzymes.
And like we said earlier, the way you understand an enzyme, or a catalyst rather, in a reaction, is that you're not changing anything at the start, you're not changing anything at the end, you're changing the path in the middle. And to get over that hump of activation energy, the whole point of it is to reach the transition state of the enzyme. So if you're comparing how well do two enzymes do against each other, comparing that transition state energy is basically comparing activation energy. Awesome. And we got it. Okay, question number 5. The active site of an enzyme E, which catalyzes a reaction X, is partially denatured. Okay, so the enzyme that catalyzes the reaction is partially denatured, not working as well. Which of the following quantities associated with X is most likely to be affected by the partial denaturation of E compared to the native form of E? Okay, which of the following quantities of the reaction is affected by the enzyme not working well? Is it the Boltzmann constant, the equilibrium constant, the rate constant, or the heat of the reaction? So the enzyme's not working as well, and we want to know a quantity that has to do with the reaction, not necessarily with the enzyme. So you said earlier that equilibrium constant is a no-no, right? Would that be the case here?
See, that's what I don't know, because we're actually, yeah, I'm thinking it's still a no-no here.
So that's good, because I immediately thought that heat of the reaction is out of the way. This is talking about kinetics. It's not talking about thermodynamics. Then the Boltzmann constant, I don't even remember what that is.
Yeah, I don't remember it at all from biochem.
So for that reason, we can just throw it out, obviously.
Yeah.
And the rate constant kind of makes sense to me. If you have an enzyme that's not working as well, then kinetically it's not working as well, it's not binding substrate as well, then it might not be doing the reaction as quickly as it was.
So real quick, I want to pause. So I remember that equilibrium constant never changes when we're talking about enzyme stuff, but let's define what that is one more time. Do you remember?
I don't.
Okay.
I need to be taught.
So, well, I don't know that I remember it specifically. I just know it doesn't change when it comes to enzymes. So what I think it is, going back to OChem and GenChem here, would be the rate at which our products are being formed just as quickly as our reactants are being reformed. So our K sub 1 and our K sub 2 would be the same as our K sub negative 2 and our K sub negative 1.
That's right.
And the enzyme wouldn't change that point.
Mm-hmm.
It might help us get there quicker, but it wouldn't change that point.
Right. Okay, that's starting to make sense. And sure enough, we were right with the rate constant. Okay. So next question. The induced fit model of enzyme binding states that which of the following molecules alters the enzyme active site to more closely match the shape of the substrate? Is it an allosteric effector, a cofactor, the substrate, or a coenzyme? Okay, induced fit model. Which of the following molecules alters the enzyme active site so that it matches the shape of the substrate? I don't want to feel like an idiot here, but is the answer the substrate?
Yeah, so when I'm thinking about this, I'm thinking about, so induced fit, meaning that some sort of change, conformational change would have to happen, like when you're catching a baseball would come into the mitt and you would close around it.
Good analogy.
Thank you. But so our other one, our lock and key, method of enzyme binding, I think of Cinderella's foot sliding into the Cinderella shoe, just perfectly fit.
Mm-hmm.
So I think you're right. I think it's just a substrate. I think allosteric effector would be something different, and then coenzyme and cofactor kind of don't even make sense.
Yeah. And we got it right.
Nice.
Awesome. Strong work, gang. Okay, next question. The active site model, also called the lock and key model.
Cinderella shoe.
Exactly. Of enzyme substrate binding differs from the induced fit model in which of the following ways?
Oops.
Is it the induced fit model holds that the shape of the active site is permanently altered by substrate binding? No. Just no. The induced fit model holds that enzyme substrate binding does not take place at the enzyme's active site. That's a slap in the face of my intelligence.
Mm-hmm.
The induced fit model holds that the shape of the substrate is irrelevant to the enzyme substrate binding. No, I would be tempted to say that, but I think that's completely wrong. D better be right, or else I'm going to sound like an idiot. The induced fit model holds that the shape of the active site is altered during the course of substrate binding. Thank you, Sal Khan, because yes, it is the right answer. All right, everybody.
So that's our baseball and mitt one, where we close around it once we catch it. And we do our chemistry, and then we release.
Yeah, because you're altering your glove.
Exactly.
Like you would alter the active site of the enzyme. Okay, question number 7?
Mm-hmm.
It's the next one, at least.
I think.
Which of the following enzyme types catalyzes the formation of a single bond between two substrates through the elimination of H2O? Isomerase, hydrolase, ligase, oxidoreductase. Okay, so I think I see a trap here immediately, because I saw hydrolase and that caught my attention, but I'm pretty sure a hydrolase is when you add water to break a bond, right?
Mm-hmm.
And isomerase, you're not using water, you're not, creating a new bond from using water. And oxidoreductase, I'm pretty sure that doesn't fit this description either. But I do know that ligase means you're connecting two things together.
Yes, and when I first read that, I read liase, and I was really confused.
Oh, yeah.
Because it's like, the answer's not here. You're right, hydrolase is using water or some other small molecule, so we can have things like a phosphorylase that a lot of people don't realize is using a small inorganic phosphate to cut a bond.
Right.
So you're right to knock that one out. Also isomerase, yep, just changing things around. And oxidoreductase, yeah, I think you were right. I think it might be ligase.
Good work. Keep it up for 2 more. Awesome. Strong, strong, strong work. All right, next question, second to last. I think it's question number 9. Enzymes undergo a decrease in catalytic efficiency in the presence of excess temperature, but can regain this efficiency once temperature returns to normal. This suggests that increased temperature does not disrupt which of the following aspects of enzyme structure. Oh, people. All right, let's read through these very carefully. Van der Waals forces, hydrophobic interactions, hydrogen bonds, or peptide bonds, better known as primary structure of a protein. Take a wild guess, people. It's peptide bonds. So before I hit, I was about to say correct, but before I hit check, am I being a little bit braggadocious, a little full of myself? Maybe. You have to be a bit of a narcissist to start a podcast, or just love the sound of your voice. Okay, as I dig my hole. So Van der Waals forces. How could that possibly be affected by excess temperature? Right off the bat, I'm not entirely sure.
Okay, so I'm the wet lab person, and one of my jobs on a daily basis is a 6-hour process called protein purification. And the form of purification that I do is using urea. You can also use a really high concentration of salt to denature the protein. And so this can be done with, changing the pH or, messing with temperature like they mentioned in the question, or using something like urea or a lot of salt to mess with tertiary folding. You can also use like beta-mercaptoethanol to get rid of disulfide bonds. Anyway, but the idea of it is that in my form of purification, I want to get rid of all of my folding except for primary structure, because once I purify it, I then dialyze out of urea and have my protein there again. But the only thing that remained constant through that whole process of denaturation, which is what they're talking about here, is primary structure.
And do you ever use temperature to disrupt things like Van der Waals forces, hydrophobic interactions? Or do you just have other chemicals that are more efficient at doing it?
So the urea is really important for disrupting hydrogen bonds, and so that helps us unfold. And then, the most stable form of I guess, conformational stability are those disulfide bonds, which we use beta-mercaptoethanol to disrupt.
Okay.
Yeah.
Take-home lesson here for everybody. Always pick peptide bonds or primary structure. Okay, final question. Let's finish it strong and hopefully not get it wrong. That rhymed. Local conditions can affect the specificity of an enzyme for its substrate, and thus the enzyme's catalytic ability. Which of the following alterations would most likely not affect an enzyme in this manner? Okay, so local conditions affect the enzyme. So which local condition would not affect the enzyme most likely? Is it increased concentration of hydrogen, increased temperature, increased concentration of hydroxide, or increased substrate concentration? So that was a bit of an audible moan that you had right there. Any
Yeah.
reason for that?
Yeah. So when they teach biochem, they, without even getting into all of the fancy schmancy stuff about tertiary structure and denaturing purifications and all of that junk, they teach that two things denature proteins, and that's temperature and pH. And so the only answer choice that leaves is increased substrate concentration right off the bat.
Yeah, it also kind of wouldn't make sense because it's the substrate
Yeah.
of the enzyme. And when you look at like a Michaelis-Menten plot, as you just boost up substrate like crazy, it's only going to increase it reaches V max, means more of the reactions happening. I'm floundering. We got it right, so it doesn't even matter what my explanation was because I was going off the rails.
So he brought up enzyme kinetics, and I'm excited to get into that. I think that's the next set.
People, if you get 100%, they throw a confetti party for you on Khan Academy. Sal, you're doing it right, my bud.